back for another video on sensory function. Again, sticking with general principles here, um, principles that apply to the sensory function more broadly. And so I want to talk about sort of the idea of processing sensory information, receiving it, and then sometimes sort of filtering out sort of peripheral sensory input. And those will all make sense in a minute here. So the first thing I want to talk about is a process called adaptation. Right? And so adaptation, well, let me back up for a second. So let's put this in context here. We have a sensory neuron, right? And if we get a stimulus at our nerve endings, right, and we'll stick with the non-special senses here. We don't have to have a receptor cell for the moment. And in those free nerve endings or encapsulated nerve endings, we're going to generate the receptor potential, right? That change in membrane potential that's caused by receiving a stimulus that matches the receptivity of that cell. In other words, if it's a receptor cell that has mechanically gated channels, then it will only sense mechanical distortion, right? Bending, stretching, pulling, whatever that might be, right? So okay, so we get our matching, our cell matches our stimulus, right? As I said before, like a mechanosensor does not detect temperature changes, right? So let's say touch arrives on your arm, right? Somebody touches your arm. The receptors in that location are going to, the receptors that have mechanically gated channels will respond, right, to that modality, and they'll generate a receptor potential. Well, if the touch was too light, then we don't reach the threshold of the voltage gated channels at the axon initial segment, and you never get action potentials. You need action potentials to convey the information into the CNS for perception. You don't get action potentials, there is no perception, right? The stimulus it's not ignored, it's never detected. So now let's jump over the threshold. Let's say our stimulus is big enough, right? And our receptor potential reaches threshold, the graded potential. Receptor potential is a graded potential. It can be bigger or smaller, right? It's big enough to open the voltage-gated channels at the axon initial segment. Off to the races we go. Now we're generating action potentials, right? Now we can talk about whether these receptors undergo adaptation, and is it complete or incomplete? So what is adaptation? Adaptation is this process of sort of adjusting the neuron, adjusting its output, its generation of action potentials when the stimulus is still present, right? And so we might have thought of ahead of time here is it's sort of like, well, as long as there's a stimulus and it reaches threshold, you're going to generate the same sort of frequency of action potentials, and sort of says, the stimulus is here, the stimulus is here, the stimulus is here, right? And go on as long as the stimulus is present. And that makes sense, except that's not how it works. Or at least that's not how it works in many receptors. There's this process of adaptation, right? And that adaptation can be complete. And if the adaptation, I'll explain the complete in a second. If that adaptation is complete, the receptors are called phasic. Or it can be incomplete, in which case the receptors are called tonic. All right, so if we have a stimulus duration here, I'll put this as a bar. In other words, the stimulus started here and ends here, and it's continuous that entire time in between, right? So time is on our x-axis here. Starts here, goes off here. It could be 3 seconds or 3 minutes or 3 days. In a completely adapting phasic receptor, what will happen is, so this would be the output of the neuron, right? In other words, the action potential generation rate. right? Because remember, back up. Information is coded in the nervous system by the frequency of action potentials. So action potentials are the language. It's the only way we can convey information, transmit information from point A to point B, right? Usually, there are anaxonic neurons, but usually we have to have action potentials. What those action potentials are conveying in terms of information is coded in the frequency and is also determined by the processing centers. Again, go back to the last video, visual processing centers only can interpret light, and auditory processing centers can only process sound, right? But okay, that's interesting, right? Because it means action potentials are the same, which are the same everywhere, right? Voltage-gated channels, right? We didn't have different versions of an action potential. All action potentials are the same, yet some action potentials can be conveying light information, and some conveying sound, and some conveying touch, right? Those labeled lines, right? Anyway, so, but the information about this stimulus is conveyed in and I'm not telling you about anything about the intensity here, I'm just telling you that it's on, right? The stimulus is there. But the information about that stimulus is carried in the frequency of the action potentials, right? And so once the stimulus comes on, if we are completely adapting, we would get an action potential. We may get another action potential, right? And right, because the stimulus is there. But if it's completely adapting, what's going to happen is it's going to stop responding. That sounds weird, right? Sort of like, we're ignoring a stimulus. Now, so let me give you an example, right? So if you put your clothes on in the morning, you put your shirt on, and you can feel the shirt going on, right? Yet all day, you're not sitting there thinking, there's my shirt, there's my shirt, there's my shirt, right? Because your skin starts to ignore that sensory input. The sensory input's still there. Cloth is still touching your skin, but it's not being consciously detected anymore because it's being ignored, right? What's the adaptive purpose of this? Well, we get an enormous amount of stimulus to process, right? External and internal. And what adaptation does is it minimizes the processing demand on your brain, right? Your brain doesn't have to all day long be sitting there going, there's my shirt, right? Plus, if you were consciously aware of it, it would drive you a bit crazy. Another good example is if you sit down in a chair and you feel this chair as you sit down, but then 5 minutes later, you're not thinking like, well, there's the chair, right? Like it's sort of like disappeared. When does the chair come back? Or when does your shirt sort of reappear in your consciousness? When you move, right? And so when you move, you sort of alter the pressure, right? Where the shirt's touching your skin, and all of a sudden you reactivate receptors. Well, very shortly thereafter, they're like, okay, good, right? So you get this new information. Oh, something changed, right? And so adaptation tells you when things change, but it doesn't tell you about things that are constant, right? And again, that we can safely ignore, right? Now, you don't want completely adapting phasic receptors in some locations. We'll come to that in a second, okay? For incomplete, oh, by the way, a lot of textbooks put a speed on phasic and tonic, basically saying that phasic are fast adapting and tonic are slow or not adapting. Speed has nothing to do with it. That's an incorrect assessment of phasic and tonic. Either one can be slow or fast. Phasic means completely adapting. It stops responding in the presence of a continuous stimulus. Incomplete is obviously going to be the opposite, right? It's going to be not ignoring the stimulus despite it being continuously present, which is what we probably would think that they should do, right? Until we learn that, we minimize the receptor input that we don't really necessarily need. So a tonic, right? And again, same stimulus path here. Line that up, right? The stimulus shows up. What's going to happen is we're going to get our action potentials. And they may slow down. They may start to not ignore, but they may minimize the input a little bit by reducing firing rate. but it never goes away, right? So incompletely adapting neurons are tonic receptors. A good example here, much to our dismay, is pain receptors, right? If I grab your finger with the pliers and squeeze, it's going to hurt. And it's going to continue to hurt for as long as I'm squeezing, right? It's never going to just completely vanish. If we're specifically, squeezing hard enough to generate, to stimulate those probably mechano receptive nociceptors, right? Remember, the pain receptors can detect a couple different things, a couple different versions of pain receptors, but right. So those are ones where the stimulus is never completely ignored, and that's for a good reason, right? You don't want to ignore pain. It would allow us to continue to use a limb, that was injured and probably re-injure it, and make things worse. Okay, that's adaptation. Let's talk a little bit about receptive fields. So receptive fields come into play when we're talking about sort of the spatial area that a receptor is sensitive to. The easiest place to think about this, and place most textbooks talk about it, is in tactile reception, in the skin. We can also talk about it in vision, and actually a couple other places. Vision's another good one though, where the light hits a particular photoreceptor, and how big that photoreceptor's field is. In other words, how broad a spatial area is it detecting, is its receptive field, right? And so the first thing we can talk about is whether sort of like the size of a receptive field, right? And so there are sort of large and small, and let's point out the difference between them. Maybe that's obvious, but point out what they're good for, right? Right? And so this is this sort of oval. Think of this as like the surface of the skin. I'll draw some little hash marks on it here. All right, if that's the surface of your skin, we could have a sensory neuron coming in here and sending nerve ending processes to this entire surface area. Same thing over here, of course, much less branching required. Right? And we'll just have this as an arrow going off to, and well, I guess what I could have done is have it synapse on another sensory neuron, right? But the idea is it's carrying information, this is the sensory neuron carrying information into the CNS. And so receptive fields can vary in size. And what does that do? Well, remember, let's say I have a stimulus here, or I have a stimulus here on this particular region of skin, this sensory neuron can't tell the difference between those two, right? This one is going to generate receptor potential and, right, possibly action potentials. This one's going to do the same thing. Its action potentials along the same region, and so that region is mapped to a particular part of your body, this whole region, and so anywhere a stimulus hits on there, it just comes from that spot, right? There's no point discrimination. Whereas when receptive fields are much smaller, if we have a receptor here or an input here, and then let's have one over here and an adjacent, right? An adjacent adjacent sensory neuron with its own receptive field. Now those two are actually closer together, and guess what? If we have this one relative to that one, they're sensed by two separate neurons, processed separately by the brain, and now we can tell the difference between those 2 points, right? And so our point discrimination is better with small receptive fields than with big ones. It's very easily demonstrated, like by using like a paper clip or something, like open up a paper clip, and have somebody, a friend, poke you in the back with, take 2 paper clips, poke you in the back, and kind of get closer and closer together with the 2 paper clips until you can't tell the 2 apart, right? You can't tell that it's not 2 separate stimuli. Do the same thing, like on the palm of your hand or on your face, and what you're going to find is that the points will be a lot closer together on your palm or your face because the neurons there, the sensory neurons there, have much smaller receptive fields. So what this means is that we get more information, more detailed information from certain regions of our body than we do from other. We don't use the middle of our back as a major, tactile sensor of the world. We use our hands and our face, right, as sort of our primary, mechanism for detecting the environment, right? For food and touch, right? Tactile with your lips, to feel the food that you're eating, that sort of thing, right? And so the receptive fields can differ in size. They can actually overlap. That gets a little complicated, but what you can have is something like this. Right, so this guy's, just make it like a funnel now. Simplify a little bit. Right, I should have overlap a little bit more, but now, right, a stimulus hitting right in the middle would actually stimulate both receptive fields, right? And depending upon which one it hits more of, or more towards the center of, it'll sort of help you with spatial determination, right? So overlapping receptive fields actually helps us with spatial location as well. So obviously the best then for spatial detection would be very small receptive fields that overlap, right? Because then you get this sort of like weighted input next to it from adjacent receptors, and they're really small, right? So the compound effect is really good spatial separation. Now, we can take this information and we can process it like this, where that information is non-converging, right? So I'll draw it separately here. So I've got 3 receptive fields here, 3 sensory neurons. And again, I'm just going to draw the output of them as like these sort of cones from down below here. If those 3 are processed separately, all along the lines of neural connections into the CNS, that's non-converging. Sorry, I'm writing a little small there, I realized. Right, non-converging. They are not coming together, okay? What that allows is high definition, right? In other words, each stimulus here versus here versus here would be detected separately. The problem is it's reasonably low sensitivity because you only have one receptor responding to a stimulus at a given spot. Now, there's another version, which is called the converging, right? And just draw receptors that look about the same in terms of receptive fields here. Draw our cones, right? But now what I'm going to do is I'm going to have the output of these synapse on another sensory neuron, probably in the CNS, or not probably, in the CNS, right? Now, if I get a sensory input here and here, well, they're converging on one neuron here, right? So I can't spatially tell them apart because it's all funneling together. But if I get a sensory input here and here simultaneously, their output onto this neuron is going to be added together. And so what happens is our sensitivity goes up, right? So converging, so let's emphasize the key points here. So converging pathways, right? We talked about this with neuronal pools, converging, right? When the neurons come together, sending more focal input to a smaller number of targets. Right, so our converging pathways, the key bit is they have high sensitivity, but low definition, low spatial separation ability, right? I call this sort of low def, high sensitivity, right? Again, because this whole region would be added together in terms of like, you wouldn't be able to tell the difference whether the stimulus is coming here or here. But again, because we're adding it together, very sensitive. Some of our photoreceptors do this. Huge amounts of convergence in certain photoreceptors. Another group of photoreceptors are much more like this. They're non-converging. So they require a big stimulus, right? Because there's no converging, adding stimuli together, but they give you really good definition, right? If you've heard of them before, cones in the eye and rods in the eye. We'll talk about that when we get to vision. The last processing mechanism that I want to talk about is one we've seen before. I think I have room. I'm going to adjust slightly just so we don't, yeah, we're right there at the bottom. And that's lateral inhibition, right? And lateral inhibition is huge in many senses. It's really important because it allows us to emphasize borders in our sensory input. Hold that thought. So let me draw. I'm going to stick with the tactile. It's really common in vision as well, inherent to vision as well. But I'm going to do, I think we can do it with tactile. It'll help. All right, so I'm going to have one here, one here. I'm going to make my cones a little shorter because I'm running out of room at the bottom here. Right, and these guys would carry their information off to, their target cell, right? Same here. So far, what I'm showing you is serial processing of non-converging pathways, right? So I just redrew this basically. But with lateral inhibition, what we have is if a stimulus hits here, then this guy will send axon collaterals over to here and over to here, right? And those are inhibitory, right? While this one is stimulating. And so what we have, again, that's determined by, by the type of graded potentials that are generated at these target cells. But so the idea here is that if our stimulus arrives here, we want this guy to respond, right? Let's think of this as the surface of the skin, right? You depress the skin. You could be, because if this pressure is enough, it could actually start to stimulate these guys a little bit as well, right? But if you want to pinpoint this and know where it's happening, it's best if we sort of shut off the adjacent pathways and focus on the one where the stimulus is most centered. And we do that by lateral inhibition, inhibiting, and think about this all the way around, not just on the two sides. And so we get this input to the CNS, but we inhibit, even though they might be getting stimulated by this, if we're depressing our skin enough. They could get some of the stimulus from this, but we shut them off to really focus on this. And so what it does is it makes it stand out. That whole idea of making a stimulus stand out is called contrast. Huge in vision. It's very important in tactile responses. The example I give in my chapter on this is if you put your finger in a glass of hot water, right? It feels hottest right at the edge, at the water-air interface, right where your finger's coming out of the water, because of lateral inhibition, right? The temperature is hot on one side, and the part of your skin that's not in the water gets shut off, right? So we make that greater contrast between the hot and the cold, right? And so we know exactly how deep your finger is into the water, more so because of that lateral inhibition, right? So lateral inhibition generates contrast, helps us separate the difference between localize a stimulus and separate it from the surroundings, right? Another quick example, vision, right? When you read and you look at black letters, right? Contrast is absolutely essential to the demarcation between where it's dark and where it's light that allows you to actually see letters on a page and read, right? Lateral inhibition makes those lines nice and crisp, which is what you want for clarity in your sensory perception. Okay, so we got adaptation. Do we want to stop responding to a stimulus over time so we can ignore it and minimize the processing need on the brain? Do we need to be aware of it and continue to generate output? That would be tonic receptors. Continually let us know that something's happening. Think pain receptors. We had a couple different ways that our receptive fields could sort of like add together or not add together, right? Non-converging, don't add together. Converging, add together. They can be big or small. That changes our point discrimination ability, right? If they're small, we get high def, right? Better point discrimination. And then lateral inhibition helping us to generate contrast. All right.